Dr. Dr. Lorna Herradura from Bureau of Plant Industry, Davao National Crop Research Development and Production Support Center, Davao City, in the Philippines. Um, may I introduce her before we go further? Yeah, Dr. Lorna complete her master PhD in University of the Philippines, Los Banos, Laguna, and Catholic University Leuven in Belgium. Currently, Dr. Lorna is the Agriculture Center Chief in the Bureau of Plant Industry Davao. She has been passionate in plant pathology, particularly on tropical fruits since 1980s. Yeah, Dr. Lorna was awarded several numbers of scholarship, fellowship, including a group award for research in 2019. Yeah. Dr. Lorna has published several numbers of international and national papers of her interest. Without further ado, please welcome Dr. Lorna for her presentation on management of bacterial disease of local Philippine banana cultivars. Please, Dr. Lorna, the floor is yours.
Thank you very much, Dr. Rosita.
Yeah.
And good afternoon, good morning, and good evening to everybody. I will be presenting the status of the management of bacterial disease of local Philippine banana cultivars. Next, please. So the presentation would just be on the bacterial diseases on banana, wherein I will focus on bugtok and dry rot, the management that is being done, and ways forward. To start with, bugtok disease, wherein the causal organism is already identified as Rostonia solana sharum, was first observed in the Philippines by Roperos et al. In 1965. So as early as 1960s, this was already reported by one of the researchers of the Bureau of Plant Industry here in Davao, which is our research center. Bugtok is actually the local term in the southern Philippines, our Bisaya dialect, to describe the infected fruit, which are discolored and hard. Bugtok would actually mean hard. So next, please. The survey that we have here, which has actually not been updated, was done in 1995. And you can see that bugtok was recorded in the upper part of the Philippines, which is in Luzon, in the Visayas, and in Mindanao. In some parts, in 1995, they were not recorded, but presently, reports have shown that they are already observed in all parts of the Philippines. Next, please. So where are they observed? We have found out that they were usually observed in plants or banana with a B genome. So these are in our cooking cultivars, the cardaba or saba, wherein this was originated in the Philippines. So we actually categorize cardaba with a triple B, identified as a cooking type, and we use this as an alternative staple crop. That's why after Cavendish, this is the most planted banana, which is our local cultivar, because now they are being exported as semi-processed, either also fresh or chips. The synonyms of this cardaba in the different dialects in our country, we call this dipig in the northern part of Luzon, In northern Mindanao, we have Kadisnon. You might identify this in Southeast Asia as Pisang Kematu in Malaysia, Pisang Kepok Besar in Indonesia, and Choy Mak in Vietnam. Also, a promising banana cultivar that we have is Mundo, which is also being affected by Bugtok. Again, this is a triple B type and also a cooking banana. Next, please. Other cultivars that were observed to be infected at the banana gene bank in our center, we have the Mundo again, Turangkog, Paadalaga, Bigihan, Hinabanico. These are actually banana cultivars. Java, so you can notice that they have all the B genomes. Gubao, Katsila, Pilipita, Maduranga, and the giant Kalakwa. Next, please. So aside from the cooking cultivars, we have a desert type that is also being infected. This is our Latundan, which is consumed in the domestic market as dessert or table banana, and also being exported in niche markets. For the synonyms of this Latundan, we call this Tundan in other parts of the country, Turdan, or Kantong. Now, in Southeast Asia, these are the Pisang Cristali in Malaysia, Pisang Raja Sere in Indonesia, Luai Nam in Thailand, Chuai Boong in Vietnam. And internationally, we call this silk or manzana. So because of the B genome, these are also being infected. Dr. Nandita actually mentioned before that she mentioned Bugtok/Moko because they have the same causal organism, and as identified in one of their publications, Mocha and Bugtok has the same sequic variety, which is sequivar 3. I would not elaborate on this, but I would just like to mention that although they have the same causal organism, their mode of transmission is different. I will not discuss on Cavendish, which is infected with Moko, but you all know that with Moko, they are mechanically transmitted, so they can be transmitted by infected farm tools and plant parts, but this is not the case for the Bugtok. For Bugtok, we can still use the followers, and they will infected, rather, the farm tools that are used in this infected Bugtok will not transmit the disease. Next, please. Next, please. What is the mode of transmission of Bugtok? This is transmitted by the insects. So this has been identified as bees, wasps, and thrips. And they are actually visiting the inflorescence, the male bud, during the opening. However, the transmission, the insects have actually not been identified in the species level. So we really have to look into this. Next, please. So what are the signs of the infected plants of Bugtok? If you incubate this, we have bacterial oozing from the infected plant parts. So you can see that this reference is still from 1995, wherein this disease was studied well as a masteral thesis at UPLB. Next, please. The signs, we have bracts covering the male bud, which are slightly rolled up, dry, and loosely attached. So this would differentiate healthy from infected plants, because in the infected plants, the bracts will not easily fall off. Next, please. These are the common symptoms on the fruits, especially on cardaba and the cooking bananas. We have the pronounced reddish brown or black discoloration, starting from the core and progressing towards the whole bulk of the fruit. You might see that in some instances, all of the hands would be infected, but in others, there are some that escape infection. So this is due to the visit of the insects during the opening of the male buds. Next, please. So other symptoms of cardaba, we can also, when you open the peduncle, or you can observe the discoloration, the black discoloration, as well as that in the corms, if they are already in the severe or advanced stages. This, you can see this in the core of the pseudostem. However, as mentioned earlier, this will not transfer to the suckers. Next, please. This is the desert type, the latundan, wherein this is also infected. This is actually, the infection is more or less severe, but sometimes you just can notice some reddish discoloration near the peel of this latundan. So generally, this is not severely infected. Next, please. After the 1995 study, the only progress that has done in the management or control is in the improvised fruit bagger. So we still do bagging of the male bud at venting stage of the inflorescence. The bagging materials that we use, we have the plastic polyethylene bags, muslin cloth, or jute bags. Or at this time, we use the Lorsban impregnated plastic bags. So how do we do it? This is the improvised fruit bagger that was developed by a state university here. We bag the still closed male bud, wherein they have improvised about a 3-meter height, so that it would be easier for the laborers rather, to bag the inflorescence. And this would be bagged for about 14 to 15 days. Next, please. The old method was actually just doing the manual bagging, wherein, as you can see, we have a steel ladder here, or sometimes they use a bamboo ladder to cover the male bud. Still, we do the 14 to 15 days bagging, or when the fruit has actually developed. Then after that, we do the budding. Next, please. Next. So this is the budding, wherein they cut the male bud when the fruits have already developed. Next, please. So aside from the budding, we can do early debudding, which is wherein they actually do not cover the male buds, but just wait for the fruits to develop. Then when they have seen that this already fully developed, they do the early debudding, which actually would not actually guarantee 100% control. Next, please. The most easier method that we actually recommend is to just do field sanitation and the lifting of dried leaves. However, since cardaba is a backyard plant, or generally it is not planted in massive hectareage. This plant is actually more of a self-supporting. Field management is not being done. That's why control of this has not really been effective, even with the recommendation of field sanitation or the leafing. Next, please. I would also like to mention another disease that we reported in 1994, which is actually the dry rot disease. You can see this was observed also in Cardaba, in backyard farms in Davao City. Next, please. The disease is similar in Bugtok, caused by the bacterium Ralstonia and the blood disease. Next, please. We just had initial studies on this, but we saw that using the DNA from infected plant materials, they have similar amplification products as that of the Bugtok when used as template. But other than that, no further studies were done on dry rot. Next, please. The symptoms on Cardaba are actually almost the same as that of Bugtok. However, when we cut open the still green but shrunken and distorted fingers, we can also observe hollow chambers where the flesh has rotted. So immature fruits are observed to stop peeling and become distorted and then turn necrotic and dry out. Next, please. So these are actually just the two main bacterial diseases that we have in our cooking cultivars and the local variety Latundan. However, we can, as mentioned earlier, this is still present in all of our plantings all over the Philippines. So what are the researches that we need to do? We still need further study and identification of insect species associated with Bugtok and dry rot disease, because we just look into the order level. Then more studies on the transmission and epidemiology of the Bugtok disease on banana cultivars with big genome. And next, please. And again, the current status on the bacterial diseases in the present millennium, because as mentioned earlier, our survey was just on 1995, but reports from technicians from different parts of the country says that this is, again, a major disease occurring in the local cultivars, especially since this is not well managed. That ends my presentation. Thank you very much.
Okay, thank you, Dr. Lorna. And well done for the successfully give an excellent presentation to us on the management of bacterial diseases of local Philippine banana cultivars. Hopefully, all of us know the difference, between Bactok, Moko and blood disease. So we have 2 minutes to Q&A. So I open for question from the floor. Yeah.
Question?
Sorry.
There is a question from Dr.
Yeah.
Chap.
Yeah. You can answer that, Dr. Lorna. Is the pulp dry rot or soft? Yeah, from Dr. Chatur.
Dr. Chatur, the dry rot in the pulp is rot. Is dry and hard. It's just like a Bugtok. It's not soft.
So it's not soft, yeah?
Yeah.
Okay, that's answered the question. We have question number 2 here. From Shazwan. Do we need to discard far away the disease plant from the field, Dr. Lorna?
If we are talking about the bugtok, without any other mixed infections, we do not need to discard it far away from the deceased, from the field, because it's not mechanically transmitted.
Mhm. Yeah. So I have a question here, Dr. Lorna, regarding the baktok. Yeah. If you can see, if you see from the slide that you share, mostly the control is from the male flower. Is the baktok can occurs before the flowering or fruiting? I mean from the early transfer the material, the banana in the field. I mean in the early.
As per the studies,
Mm.
the transmission occurs when the flowers opens and the insect vectors visits in the early morning, wherein incidentally these insect vectors might carry the bacteria. So before these, they are not infected. So the transmission is from the upper portion from the male bud down to the form.